Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Sarathi Vijay Boddapati from Catalent.
Thank you, Leah, for the introduction. And as Leah said, I'll be talking about formulation and characterization strategies for gene therapy today. Just to give you a brief introduction of myself, I've been at Catalent and I've been with the company for approximately 2 years, running the formulation development group here. To give you an overview of the Catalent company itself, our mission here is to develop and supply products that help people live better, healthier lives. And you might know Catalent as a worldwide company that works with several different molecules that go into oral formulations, biologics, respiratory and ophthalmic formulations, and also of late into cell and gene therapy. That's where I'll be concentrating most of my talk on. Catalent itself is spread out all over the world with approximately 15,000 members, and it is into different technologies that work with development, delivery, clinical logistics, and into clinical and commercial manufacturing as well. And as I mentioned, I'm working for cell and gene therapy. The gene therapy aspect of Catalent is based in Maryland, and that's where we are located. And predominantly what we work with is related to AAV vectors. AAV vectors, as is listed here, these are the primary vectors that are used for gene therapies. They are quite non-pathogenic in terms of their properties, and that's why they make a great tool to deliver DNA into a cell. They are made by including 3 plasmids, infecting cells with 3 plasmids that include the gene of interest. And then those are purified through several different steps in formulation, through process development and formulation. The shell itself is made up of the proteins VP1, 2, and 3 that then encapsulates the gene of interest. That's the DNA payload. And being non-replicating, that's another benefit for the AAVs. They're non-replicating, and that's why they cannot integrate into the host genome. And the VP1, 2, and 3 proteins are usually in a ratio of 1 to 10. And as different serotypes accumulate different VP1, 2, and 3, amino acids differences occur, and that leads to different serotypes being formed. And these different serotypes then are able to target different tissues. And that's a big advantage of the AAVs as well. A couple of other advantages with regards to AAV is that they are highly stable in their natural form between a wide range of pHs, approximately 3 to 9. They are approximately stable for a couple of hours at least at room temperature, and higher temperatures even going up to 40 C or 50 C. They are stable up to 2 hours. That makes them a great tool for delivering the genes of interest. And then the other thing that we talk about while we are working with AAV vectors is their ability to package the genes. One can view the size of the gene that can be encapsulated as a positive or a negative based on the type of gene that you're targeting. It is restricted to 4.7 kb, which means that anything larger has to be split up and put into 2 AAV viruses that then get spliced when they're delivered together. And there is a few products that are coming out into clinical trials using that kind of a approach. So with regards to formulation development, the pie that you see over here is what we like to see the scope of all formulation activities as. And this slide itself is modified from what it looks like for a biologic, for a classical formulation work that goes into protein product development. And you'll see in blue is where we do the core of the work, formulation development and product characterization. Here's where we take the molecule from the preclinical stages, try to understand the molecule through the characterization studies that we perform here, and then try to also understand the degradation mechanisms that might be affecting the molecule, and then work towards preventing those degradation propensities. And then in green, you will see that we are trying to develop the final image of the product, how the clinic will handle it, how a subject will see it as it is administered. In their case, this is where the drug product development and operation comes in. Heavily involved in this is freeze thaw characterization. The drug substance, obviously, for AAV is kept frozen. The drug product even is kept frozen. This is where we would study those kinds of things. We would study the freeze and thaw characteristics of the material as it is used and as it is made from drug substance into drug product. And then finally, the last one here is what we look at as pharmaceutical packaging and combination product development. And this is where we start to look into the container closures, the stoppers, the devices that go into use in the clinics. And in the case of AAVs, the container closure itself has been CZ vials predominantly, and that's because the product itself is frozen. And moving along to my next slide, here's where I will concentrate a little bit more on the activities as a comparison between the AAV virus vectors and the proteins and peptides. You can see on the left is AAV viral vectors. You'll see most of the categories on this AAV side of things are similar to what you will see on the right as well. They are just in a different order. This is laid out in the order in which we expect to see activities occur in the formulation realm. So for example, pre-formulation development, since the product is stored -80 under frozen conditions, the drug product is filled into a vial and put into a freezer, and it's taken out when the dosing needs to occur. Freeze thaw compatibility, and then what we would see as higher temperatures than normal storage, -40 C even and -20 C as higher temperatures. That's what we studied during pre-formulation activities. And just to contrast this to the protein side, on the protein side, during pre-formulation development, we'd be straight away looking at screening buffers, excipients, and such things. Whereas on the AAV side, we are not front loading that on the AAV side. With regards to clinical device usage, which is our next bullet here, there is a heavy usage of tubing sets and other devices, since almost all AAV products are handled in a clinical setting. This becomes an important activity. Drug substance optimization, here is where we integrate a little bit with our process development colleagues and continue to look into buffers, process conditions, compatibility with the different buffers that they use. And then I had mentioned characterization as an early activity that we do, biophysical characterization, using some of the techniques that are routinely used for proteins. That's something we would do as well. Packaging and shipping studies, these have not grown in prominence as of today for AAV, but we do expect that as we move forward and as more products come into prominence, we would see that we would need these studies. And then lyophilization as on today may not be a feasible or a viable option for a product that is only looking at thousands of vials. It will come into prominence if we do need to see something where it goes into the hundreds of thousands of vials, where storage becomes an issue and freezer space becomes an issue. On my next slide here is a little bit more of an overview of the formulation activities itself. Biophysical and chemical characterization, we are looking now into the techniques that have been used for proteins and peptides. Biologics, these are DSC, DLS, DSF, AUC, and HPLC. AEC and HPLC we know is used on a routine basis for AAV characterization, especially with the empty fulls. DSC and DLS have also been used. DSF is becoming a new tool that's coming on as a big characterization tool for AAV materials as well. Forced degradation conditions, I mentioned this in one of my previous slides. We are now starting to look into degradation mechanisms. The conditions that we look into listed here: temperature, pH, the processing conditions during drug substance purification, drug product formulation is what we look into here. And then oxidation is one of our favorites in terms of forced degradation. Once we have some data with characterization and forced degradation, we start looking into the buffers and excipients. And again, as I mentioned, in our current format, this is restricted to pH between 7 and 8, and that being historic as well as the delivery of the molecule into regions that are quite sensitive to osmolality. And that's why it's restricted to 7 or 8. Our focus here will also be into salts, detergents, sugars, amino acids, those kinds of things. There is a whole bunch of literature that suggests that the osmolality plays a big role with one of the predominant mechanisms of degradation, which is aggregation. Salts like magnesium chloride, sodium chloride, things like that, and looking at osmo for those materials. And then finally, we do some stability in the formulation side of things. And this stability is predominantly to confirm that the formulation that we have come up with, that we have selected, is suitable for its intended use, and it is able to keep the product stable in its intended storage. Again, this is -80 as of today. And then I had mentioned DLS and DSF as two techniques that are now evolving. DSF predominantly is evolving as a key technique in AAV characterization. Here's some examples that I'm showing. And from a DSF instrument, like an Unchained Labs, the instrument itself is called Uncle, or from a nanotemper equipment, we can get multiple data outputs from one single equipment. Previously, we used to have to use DSC to get the TM. We used to have to use DSF, fluorescence-based measurements, again, to get some aggregation propensity measurements, and then look at zeta sizer or some kind of experiment, DLS equipment to get the size for this. Now, with the advent of these kinds of equipment like the Uncle, we are now looking to get all of this data from one single equipment. And more importantly, with a very, very small quantity of volume, in some cases approximately 10 microliters. So compare that with a classical DSC, where you would need higher than 500 microliters of volumes to 10 microliters of volume, and getting all this information. And just to point out from the data itself that's shown on here, we compared two different AAV serotypes on here. And we are looking at the Z average in the table for DLS, and we are looking at 13.6 for one of the AAVs and 42.8 for the other. And as this is heated through up to 95 degrees Celsius, we see that they change different rate, different final endpoint that you see for the two AAVs. That similar thing is reflected on the picture in the right for AAVX, and these are aggregation curves that were obtained on the Uncle. You see that there is a nice flat line for AAVX up to the point of approximately 70 degrees Celsius, and then it starts to aggregate from that point onwards. Whereas for AAVY, you see that it is already starting to show some propensity for aggregation in the low 30s, maybe due to its nature of the buffer that is suspended in, or the way it was purified. That's something that we have not investigated for this material, because this was a commercial material that we purchased. And that's what we are seeing on this slide. So the utility of the equipment is kind of what is shown here, and that's something that we intend to use as we move forward in our development and activities further. And then we talked about device compatibility a little bit in our earlier slide. And this is one big thing that is used for almost all of the gene therapy clinical trials, as well as for the intended final products. Considering that there are several sites of injection, brain, retina, muscle, ear, etc., there is the use of infusion pumps, and there is the use of different kinds of syringes, cannulas. A couple of examples are shown on the right with the BD system and something from Smith's Medical. These are pumps that are used to deliver the infusion, deliver the dose through the cannula. And you can see the cannula itself is quite a narrow gauge, narrow bore. And what that does is that it creates resistance when the injection is happening. And that's the reason why we have to look into infusion time, confirm the accuracy at which dosing occurs, compatibility of the AAV with the components, with all the components that go into making all the products that the product encounters, and then also looking at hold times so that when the clinician or the nurse that is administering these materials, they have enough amount of time between preparing the sample, getting the patient ready, and getting to the point where the infusion actually occurs. And then, looking at stability of that product under those conditions is quite critical. So that's the kind of list that we have prepared here and shown. And obviously, this is one key portion that we've been working quite extensively on. And then finally, just to summarize the activities, formulation development, we do believe is quite key in terms of AAV as a product. As of now, formulation activities are limited, and that's because of the frozen storage condition. That's because the number of drug product vials that we are looking at making is in the low thousands to mid tens of thousands, not higher than that. Once we start growing beyond that stage, where the number of subjects is higher, where the amount of material that we produce as drug product, we do believe that formulation will become more critical, and even starting to look into the possibility of having formulations that can be stored outside of the freezer, or even in the freezer that's more easily available. For example, a -20 as opposed to a -80. And then, as I showed you in my slides, we are applying a lot of the knowledge that we have gained from biologics to the AAV, looking at formulation screening and development, developing new tools, adapting existing tools, and bringing on new tools as we go on. And then we expect that as the field grows, by front-loading all of the formulation activity works, we have a better ability to impact the future of new products that come into the market. And on this last slide here is my contact information. Feel free to reach out if you have questions, and I'll be happy to take any questions now.
Great, thank you. So the first question is, how do the process development activities mesh together with FD activities?
So that's so obviously the process development is where formulation usually gets their materials from, and so it is in our best interest to ensure, along with formulation development, along with the process development colleagues, to make sure that the material that we receive so that we can continue our work is of good quality. There is obviously a good interaction at Catalent between the formulation and process development folks, especially when process development is occurring in the later stages of purification. Having said that, the AAV process development itself is a platform, if you will, considering that most of the steps remain the same for all the AAVs. You're looking into tweaking conditions for different serotypes or different companies based on their desire product profile. So that's where we do get involved with process development a little bit and start looking into optimizing or preventing aggregation across some of the columns, optimizing some of the buffers that we then can work further on in formulation development.
Okay. And are there any platform studies on buffer excipient suitability with AAV serotypes?
The short answer to that would be a yes. And if I had to elaborate a little bit on that, we do want to develop formulations that are platform. And as I mentioned, currently the buffers that we are looking at fall in the range of 7 and 8. We would like to expand that further to at least 6 to 8, considering that some of the buffers that have the most impact, especially on biologics, have been around the 6 to 8 range. So in terms of developing a platform, I think that's where we would focus our efforts on, in trying to expand the pH range, trying to expand on the excipients that we would try to include going forward in the formulations. And this would obviously be dependent on gathering the information from various techniques that give us the characterization information as well as the degradation propensity information, and then trying to prevent those. In the end, we would probably want to have a list of buffers, a list of excipients that we can assign to a serotype. Now, the challenge to having a platform itself is being able to get material, considering that the yields for AAVs are quite limited and material availability is quite limited. If we had to develop a platform, we would have to generate quite a lot of material to work with, unless we are using techniques like the DSF that uses low quantities of materials. Another challenge that goes into consideration when looking for a platform is the serotypes. There is approximately 13 serotypes. There are at least 7 to 8 different serotypes that are being investigated in the clinic. So if we had to develop a platform, we can do that starting off with one of the AAVs and then applying it across the different serotypes. That's one approach to take.
Okay, well, it looks like we're running out of time for questions. We have time for just one more question, but if we haven't got to your questions, go ahead and enter any additional questions. We'll be passing any question that we haven't answered directly on to Sarathi, and he can follow up with you. So the last question for the webcast is, are there any techniques to analyze empty versus full capsids during formulation development stages?
The empty versus full during formulation development plays a less important role, in my opinion. There have been some literature that suggests that the presence of empty may have an impact on the aggregation propensity or on the stability, but there is nothing proven there. That said, process development is usually where the empty full assay becomes more important. AUC is the current gold standard, but obviously AUC is needing a lot of volume in terms of testing. Therefore, at Catalent, we have been looking into adapting the ion exchange assays. There is a few publications that talk about ion exchange, HPLC, as a method looking at empty full. We have been adapting that method and trying to run that for the different serotypes and trying to provide that as a tool for our process development folks, especially, to look into empty and full and trying to understand changing the different process conditions and the impact of that on empty full during purification.
Okay, thank you, Sarathi. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a email providing us a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.